Live from room 1242 in Highland High School, it's Hawk Science time, featuring your lab, Beanie Babies, and the laws of probability, with your host, Mr. King. Okay, so today we're going to be playing with probability. We're going to be using brown paper bags and beans to represent the passing of genetic traits and different alleles. So at this point, everyone should be looking at their lab procedures. I have a list of materials up above, and I will be providing you with that shortly. But looking at the procedures, we can see that we are going to be taking the beans and placing them into each bag and simply just pulling them out simultaneously to simulate the birth of a new child originating from our parent generation. Today, you're also going to need this data table. Okay, just a quick review. An allele is one of two forms of a gene, and we're going to try to predict today what alleles the offspring are going to have from our parent generation. However, there are two kinds of alleles, dominant or recessive. If a dominant gene is present, it's going to be expressed over the recessive allele, and we're going to see that in the form of a phenotype. So we can have a dominant phenotype. However, the genotype for this case is going to be a capital letter. I'm going to use R. You may also have the recessive allele, which produces the phenotype only when paired with the recessive allele. So you may need to have two recessive alleles to express that trait. I'm going to use a lowercase r for today's lab. And one more vocab review. Hetero means different, homo means the same. Okay, so let's start here. We're going to have two true breed parents. Grandma is going to be homozygous recessive, and grandpa will be homozygous dominant. And they are going to breed. So let's predict the outcomes here using a Punnett square. Let's put grandpa on the top and grandma on the bottom. And if I fill this out, I can see that my big R's are going to meet up with my little r's. So grandpa is going to give one allele and grandma is going to give the other. And I get 4 out of 4 boxes heterozygote. So my ratio becomes 1, and I have a 100% likelihood of having an offspring being heterozygote. So I'm going to take some white beans, 40 to be exact, and I'm going to count them into grandma's bag. I've pre-counted my beans, so I'm going to dump them in the bag. I'm going to add 40 red beans, representing the dominant allele, to grandpa, in which I've already counted them out, so I'm going to add them quickly to our bag. Now, simultaneously reaching in each bag, I'm going to pull out the offspring's alleles. You can see right off the bat, I'm getting an abundant amount of heterozygote alleles. This is exactly what we just predicted. Okay, now for F1 generation. So we are going to divide up the beans, and we are going to have two heterozygote parents. If they are going to mate, we can fill out another Punnett square and predict the outcome. So let's cross two heterozygotes, and you're going to see we're going to have a big R, a little r, big R, a little r, big R, and then two little r's. So our outcomes, we can have homozygous dominant, heterozygote, or homozygous recessive in the form of a 1 to 2 to 1 ratio, meaning that I have a 25% chance of having an offspring homozygous dominant, 50% chance of having an offspring heterozygote, or a 25% chance of having a homozygous recessive. I'm going to divide the 40 pairs of alleles now between our F1 generation, mom and dad. So each bag should have 20 red and 20 white beans, being completely heterozygous. Once I've done this, I can start over again, reaching simultaneously into each bag and pulling out our random paired alleles. This is a random event happening. You can see right off the bat, I'm getting different alleles every time I reach in the bag. Now make sure not to peek, this should be completely random. We're hoping to see the 1 to 2 to 1 ratio that we just predicted before. Now is a good time to fold up your bags and to divide out the beans one last time, so that'll make for an easy cleanup job. Okay, we're almost done. Now let's take our data and add it to our team data sheet. Make sure to get every group's data so we can get the total amount. While you are waiting for groups to finish, please start the discussion questions. Since we are going to show a comparison, we need to also use a bar graph to finish this lab.